In this section, we will be looking at microbial genetics. The genome is the entire genetic component of the cell. The gene is the sequence on the genome that codes for a specific polypeptide or RNA. The genetic material, both RNA and DNA, is composed of base pairs of specific pairs of nitrogenous bases that pair as follows: G and C, or guanine and cytosine; A and T, or adenine and thymine; or A and U, adenine and uracil. The strands of DNA have two specific ends, specified as the 5' ends and the 3' ends. The 5' end terminates in a phosphate group attached to the 5th carbon. The 3' end terminates with a hydroxyl group bound to the 3rd carbon. The strands are said to run anti-parallel in that one strand runs from 5' to 3' and the complementary strand runs from 3' to 5'. There are slight differences in the prokaryotic and eukaryotic genomes. Both have chromosomes, which are organized forms of DNA with their associated protein. Bacterial or prokaryotic chromosomes are typically circular, and there is usually one or two of them. The localized region where the chromosome is found is the nucleoid region. Prokaryotes also have plasmids, which are small circular DNA molecules which replicate independently of the chromosome. The F, or fertility plasmid, contains the instructions for bacterial conjugation. The R, or resistance plasmids, carry genes for resistance to one or more antimicrobial drugs or heavy metals. Bacteriocins are factors or toxins produced by the genes that are designed to kill the same or similar species without the factor as a way to kill the competition. Plasmids may convey virulence with the instructions for enzymes or toxins to make them pathogenic. The eukaryotic genome is surrounded by a nuclear envelope consisting of a double membrane surrounding the genetic material. Globular proteins called histones are used in packaging the DNA into chromosomes. The DNA is negatively charged, which attracts it to the positive charged histones. The 10 nanometer beads of DNA will wrap around the histones to form nucleosomes. The nucleosomes will clump with a protein to form 30 nanometer diameter fibers dispersed throughout the nucleus as chromatin. When in the form of a chromosome, the single leg of the genetic material is the chromatid. There is also extranuclear DNA found in the mitochondria, chloroplasts, and plasmids. When DNA replicates, it carries the energy for synthesis within the high-energy bonds of the molecule itself and does not need further energy from the cell. Replication is semi-conservative because of the new DNA being composed of one original daughter strand and one new strand. The replication of DNA is broken into 3 parts: the initial process, the synthesis of the leading strand, and the synthesis of the lagging strand. Here we have an animation on DNA replication. So here we can see the DNA, or the ingredients in this process in the cell. You can see the helicase splits the DNA molecules apart. The RNA primase inserts a starter of RNA nucleotides at the initiation point. The DNA polymerase binds a complementary leading strand of DNA nucleotides starting at the 3' end of the RNA primer. In humans, approximately 80 nucleotides are added per second. In archaea bacteria, the rate of synthesis can reach 1,000 nucleotides per second. RNA primase attaches more RNA primer in the remaining gaps, yielding a series of Okazaki fragments, or the lagging strand. The initial process begins at a sequence called the origin. A DNA helicase enzyme will unzip the DNA and expose the nucleotides in a replication fork. The DNA polymerase binds the strand and always adds nucleotides at the 3' end. The leading strand is synthesized continuously as a single long chain of nucleotides running 5' to 3'. The lagging strand is synthesized in short segments that are later joined. Here we have a short clip of A Link to DNA, The Secret of Life.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called Helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
When synthesizing the leading strand, the enzyme primase will synthesize an RNA primer that is complementary to the DNA template. This will provide the 3' hydroxyl required by the RNA polymerase. Triphosphate deoxyribonucleotides hydrogen bond to the complements of the parental strand. DNA polymerase covalently joins to the parent strand with energy from triphosphate deoxyribonucleotides and adds nucleotides at a rate of 500 to 1,000 per second. There may be errors at this rapid rate, so DNA polymerase proofreads the strand. About 1 in 100,000 nucleotides are mismatched, but after DNA polymerase proofreads, the error rate decreases to about 1 in 1 billion base pairs. Finally, the enzyme replaces the RNA primer with DNA. In synthesizing the lagging strand, primase will also synthesize the RNA primers. The nucleotides will also pair with the complements. DNA polymerase joins the nucleotides and proofreads. Because there must be a 3' hydroxyl, these are synthesized in discontinuous segments known as Okazaki fragments. Each fragment is about 1,000 nucleotides and requires a new primer. Another type of DNA polymerase replaces the RNA primer of the Okazaki fragments with DNA and further proofreads. Finally, DNA ligase joins the Okazaki fragments. Here we have a DNA replication animation from Wiley.
DNA replication is the process whereby an entire double-stranded DNA is copied to produce a second identical DNA double helix. The objectives of this exercise are to understand the functions of the proteins responsible for DNA replication, including helicase, SSB protein, primase, the sliding clamp, DNA polymerase, RNase H, and DNA ligase, and to understand why the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously. DNA replication is an intricate process requiring the concerted action of many different proteins. The replication proteins are clustered together in particular locations in the cell and may therefore be regarded as a small replication factory that manufactures DNA copies. The DNA to be copied is fed through the factory, much as a reel of film is fed through a movie projector. The incoming DNA double helix is split into two single strands, and each original single strand becomes half of a new DNA double helix. Because each resulting DNA double helix retains one strand of the original DNA, DNA replication is said to be semi-conservative. DNA replication requires a variety of proteins. Each protein performs a specific function in the production of the new DNA strands. Helicase, made of 6 proteins arranged in a ring shape, unwinds the DNA double helix into 2 individual strands. Single-strand binding proteins, or SSBs, are tetramers that coat the single-stranded DNA. This prevents the DNA strands from reannealing to form double-stranded DNA. Primase is an RNA polymerase that synthesizes the short RNA primers needed to start the strand replication process. DNA polymerase is a hand-shaped enzyme that strings nucleotides together to form a DNA strand. The sliding clamp is an accessory protein that helps hold the DNA polymerase onto the DNA strand during replication. RNase H removes the RNA primers that previously began the DNA strand synthesis. DNA ligase links short stretches of DNA together to create one long, continuous DNA strand. Let's look at the steps of DNA replication in more detail. To begin the process of DNA replication, the 2 double helix strands are unwound and separated from each other by the helicase enzyme. The point where the DNA is separated into single strands and where new DNA will be synthesized is known as the replication fork. Single-strand binding proteins, or SSBs, quickly coat the newly exposed single strands. SSBs maintain the separated strands during DNA replication. Without the SSBs, the complementary DNA strands could easily snap back together. SSBs bind loosely to the DNA and are displaced when the polymerase enzymes begin synthesizing the new DNA strands. Now that they are separated, the 2 single DNA strands can act as templates for the production of 2 new complementary DNA strands. Remember that the double helix consists of 2 antiparallel DNA strands with complementary 5' to 3' strands running in opposite directions. Polymerase enzymes can synthesize nucleic acid strands only in the 5' to 3' direction, hooking the 5' phosphate group of an incoming nucleotide onto the 3' hydroxyl group at the end of the growing nucleic acid chain. Because the chain grows by extension off the 3' hydroxyl group, strand synthesis is said to proceed in a 5' to 3' direction. Even when the strands are separated, however, DNA polymerase cannot simply begin copying the DNA. DNA polymerase can only extend a nucleic acid chain, but cannot start one from scratch. To give the DNA polymerase a place to start, an RNA polymerase called primase first copies a short stretch of the DNA strand. This creates a complementary RNA segment up to 60 nucleotides long that is called a primer. Now DNA polymerase can copy the DNA strand. The DNA polymerase starts at the 3' end of the RNA primer and, using the original DNA strand as a guide, begins to synthesize a new complementary DNA strand. 2 polymerase enzymes are required, one for each parental DNA strand. Due to the anti-parallel nature of the DNA strands, however, the polymerase enzymes on the 2 strands start to move in opposite directions. One polymerase can remain on its DNA template and copy the DNA in one continuous strand. However, the other polymerase can only copy a short stretch of DNA before it runs into the primer of the previously sequenced fragment. It is therefore forced to repeatedly release the DNA strand and slide further upstream to begin extension from another RNA primer. The sliding clamp helps hold this DNA polymerase onto the DNA as the DNA moves through the replication machinery. The sliding clamp makes the polymerase recessive. The continuously synthesized strand is known as the leading strand, while the strand that is synthesized in short pieces is known as the lagging strand. The short stretches of DNA that make up the lagging strand are known as Okazaki fragments. Before the lagging strand DNA exits the replication factory, its RNA primers must be removed and the Okazaki fragments must be joined together to create a continuous DNA strand. The first step is the removal of the RNA primer. RNase H, which recognizes RNA-DNA hybrid helices, degrades the RNA by hydrolyzing its phosphodiester bonds. Next, the sequence gap created by RNase H is then filled in by DNA polymerase, which extends the 3' end of the neighboring Okazaki fragment. Finally, the Okazaki fragments are joined together by DNA ligase, which hooks together the 3' end of one fragment to the 5' phosphate group of the neighboring fragment in an ATP or NAD dependent reaction. We are now ready to review the steps of DNA replication. The process begins when the helicase enzyme unwinds the double helix to expose 2 single DNA strands and create 2 replication forks. DNA replication takes place simultaneously at each fork. The mechanism of replication is identical at each fork. Remember that the proteins involved in replication are clustered together and anchored in the cell. Thus, the replication proteins do not travel down the length of the DNA. Instead, the DNA helix is fed through a stationary replication factory, like film is fed through a projector. Single-strand binding proteins, or SSBs, coat the single DNA strands to prevent them from snapping back together. SSBs are easily displaced by DNA polymerase. The primase enzyme uses the original DNA sequence as a template to synthesize a short RNA primer. Primers are necessary because DNA polymerase can only extend a nucleotide chain, not start one. DNA polymerase begins to synthesize a new DNA strand by extending an RNA primer in the 5' to 3' direction. Each parental DNA strand is copied by one DNA polymerase. Remember, both template strands move through the replication factory in the same direction, and DNA polymerase can only synthesize DNA from the 5' end to the 3' end. Due to these 2 factors, one of the DNA strands must be made discontinuously in short pieces which are later joined together. As replication proceeds, RNase H recognizes RNA primers bound to DNA templates and removes the primers by hydrolyzing the RNA. DNA polymerase can then fill in the gap left by RNase H. The DNA replication process is completed when the ligase enzyme joins the short DNA pieces together into one continuous strand.
And that is a summary. There is also a link to this website in Angel, so you can go through and practice answering the questions yourself. Synthesis is usually bidirectional and proceeds in both directions from its origin. The origin is not necessarily at the end and may be in the middle, allowing this to happen. Sometimes methylation occurs where a methyl group is added to one or two bases that are part of a specific nucleotide group. This can help control genetic expression by turning off genes or turning them on. Methylation may also play a role in initiation of DNA expression. Viral DNA is not methylated, so it helps protect against viral infection and also repair DNA.